A very good morning, respected teacher. Myself, Nrupshambhu Sarnobar, roll number 98, and today I'll be presenting a seminar on the topic oxygen hemoglobin dissociation curve. So, in today's seminar, we'll cover the topic transport of oxygen, role of hemoglobin in oxygen transport, shape of the curve, factors affecting the standard dissociation curve, that is shift to right or left, the Bohr's effect, and P50 and its significance. So first of all, oxygen is mainly transported in the blood in two forms. First one is dissolved form and next is in combined form with hemoglobin. In dissolved form, around 3% of the oxygen is transported, whereas 97% of the oxygen is transported in combined form with hemoglobin. The presence of hemoglobin in RBC allow the blood to transport 30 to 100 times as much as O2 that is transported in the dissolved form. And approximately 0.3 ml of O2 per 100 ml of blood is transported in dissolved form, whereas the remaining is transported as combined form with hemoglobin. So now what is the role of hemoglobin in this transport of oxygen? As we all know, about 97% of oxygen is transported from the lungs to tissues in combined form with hemoglobin. Oxygen combines with hemoglobin to form oxyhemoglobin. The combination is reversible. A hemoglobin chain has 4 heme prosthetic groups and each heme group has 4 iron atoms. So each of these atoms bind loosely with one O2 molecule. So one hemoglobin can carry 4 O2 molecules. The binding of O2 is loose and reversible. And in low PO2 conditions, the oxygen is dissociated and released from the hemoglobin. So now let us talk about oxygen dissociation curve. The oxygen dissociation curve demonstrates a progressive increase in the percentage of hemoglobin bound with O2 as PO2 increases, which is called as percentage saturation of hemoglobin with oxygen. The curve shows a sigmoid shape. There is a steep rise in the curve due to the cooperative binding kinetics. And as the curve reaches saturation, there is a plateau which is formed. The curve reaches the plateau as about 15 gram of hemoglobin in 100 ml of blood combines with 20 ml of O2 at 100% saturation. And when this saturation occurs, the plateau is reached. Let us look at the factors that affect the standard dissociation curve. The strength with which oxygen binds to hemoglobin is affected by several factors. These factors either shift or reshape the oxygen hemoglobin dissociation curve. These factors include pH, temperature, PCO2, 2,3-BPG, H+, etc. The curve is either shifted to the left side or to the right side of the graph. The shift in the dissociation curve. Now, the control factors such as temperature, 2,3-BPG, PCO2, and acidity can cause either right or left shift. The increase in temperature causes a shift to the right side. The increase in 2,3-BPG in PCO2 and in H+ also cause a right side shift of the curve. And it mainly occurs in the tissue region, and the dissociation of oxygen from hemoglobin takes place over there. When there is a decrease in the factors that affect the curve, such as decrease in temperature, decrease in 2,3-BPG, decrease in PCO2, or decrease in H+ concentration, there is a left shift, and oxygen is combined with hemoglobin over there. And it occurs mainly in the alveoli region. Next, let us look at the Bohr's effect. Bohr's effect is a phenomena first described and discovered by Danish physiologist Christian Bohr. It describes how hemoglobin's affinity for oxygen decreases as blood acidity keeps on increasing due to rising carbon dioxide level. The Bohr's effect refers to the shift in the oxygen dissociation curve caused by the changes in the concentration of carbon dioxide or pH of the environment. It causes a right shift in the oxygen dissociation curve. As the acidity is increased, the concentration of H+ ions is increased. Therefore, it causes a rightward shift. The oxyhemoglobin dissociates at tissue level to provide oxygen to the tissues. So what happens in the tissues is there is presence of an enzyme carbonic anhydrase which converts CO2 and H2O to form H2CO3. It further converts H2CO3 to form HCO3 minus and H+, causing a rise in the H+ concentration. Due to this rise, the curve is shifted to the right side in the tissues. Now, P50 and its significance. What is P50? So, P50 or the partial pressure of oxygen at which hemoglobin is saturated. It is a key indicator of hemoglobin's oxygen affinity. Hemoglobin is 50% saturated at this level. It reflects how readily hemoglobin binds and releases oxygen, which is crucial for tissue oxygen delivery. A lower P50 means hemoglobin has very high affinity for oxygen and it binds to the oxygen, such as in the alveoli or in the lungs. And a higher P50 indicates very low affinity, leading to an easier release of the oxygen when in the tissues. around the normal value for P50 is about 27 mm of Hg. So these are the references from where I took the images and the information. They are Geiter and Hall Book of Physiology, AK Gen, Wikipedia, and few other sites. Thank you.